thanks everyone who's still here at the end of the day. We've got a final talk of the day by Barbara Osimani about statistical evidence in strategic environment.
Thank you. Thank you, everybody, for having me. I'm sorry not to be there. I hope next time. And yeah, thank you for the great workshop. All the topics are very interesting, and I hope that at the end of the day, I won't be too annoying for you. So just to relieve you, this is not a statistical talk. It's just a talk about how uncertainty can be exploited in strategic interactions in the sciences, and especially in medicine. So let's dive in. Oh, it doesn't work. Doesn't work. Okay, yes. So science has been framed by statisticians as a game against nature. I mean, this was valid until science was practiced by, let's say, noble people or people that didn't have any economic interest in delivering research, mainly reputational interest, okay, but as science was not framed as it is now. It seems, however, that we are not only playing games against nature. Evidence collection, interpretation, and disclosure is embedded in a framework of strategic interactions among diverse players. Strategic means that these players are aiming to maximize or optimize their own welfare as a function of their specific preference settings, interests, prior knowledge, it goes on by itself. Epistemic capacities and virtues, pragmatic roles, and risk attitudes. So there is an implicit awareness, or more or less explicit, depending on the domain we are dealing with, of this state of affairs. And this may explain a series of phenomena in the current scientific practice. So the institution of regulatory agencies devoted to the control and approval of technologies. We have talked about pharmaceutical regulation, for instance. Until now, there are, of course, other agencies. Evidential standards developed by the scientific communities. We have talked about evidence hierarchies, publication standards, and the emergence of various crises and related attempts to reform science. I have to run because otherwise the slideshow goes before me. So there is this information asymmetry, which is at the heart of the scientific enterprise, but that combined with exogenous interests that divorce this asymmetry from the search for truth pervade science in the scientific environment in the current days. So the out of the talk is, this is medicine a lemon market? And what is a lemon market? And then the statistical reforms that have been proposed until now to solve a little bit these kind of problems I was speaking in the introduction. A strategic view, deregulation, and then if we have time, the density of evidence as a problem. So what are lemon markets? Lemon markets are those markets where there is an information asymmetry between seller and buyer. And this information asymmetry may incentivize the seller to deliver fake signals. And then in the end, the market may fail because of these incentives to sell products which are lower and lower the average, gradually lower and lower. So this can be cars, houses, but maybe also healthcare. So for instance, we can see here a lemon house, a very beautiful house where it rains down from the roof. Okay, is healthcare a lemon market? Can it be? In what conditions? Medical products are so-called, maybe you are seeing also these kind of things, I don't know. But, medical products are so-called credence goods, that is, products for which the consumer, the medical community, patients, and public health system, in a more general sense, also the regulators, cannot evaluate the quality prior and often not even after consumption. So there is this information asymmetry. Doesn't go on, I don't know why. Okay. Medical decisions are intertwined with pressing ethical dilemmas, where the good at stake can be the same for all alternative actions, so that is health. The disease is weighed against the possible side effects of the treatment. We saw it in the COVID case, for instance, very, very pleonastically. Medical decisions are characterized by high stakes, both in terms of physical, psychological, existential, and financial costs, both at the individual and collective level. And producers of medical knowledge, for instance, the pharma industry, but also the academia, often have vested interest in the research outputs and dissemination, leading them to engage in strategic behavior, which obviously exploits all of the above. So we have questionable research practices, scientific misconduct, fraud. What are the institutional instruments that have been developed in order to counteract this explosion of uncertainty? First of all, deontological norms and evidential standards, as we saw, these reproducibility requirements, evidence hierarchies, so RCTs are better than observational studies, and so on and so forth. Monitoring systems, for instance, the regulatory authorities, or bias detection tools. We have a lot of meta-science studies now trying to detect bias in very different forms. Statistical decision rules, for instance, Bayesian reciprocal frequency statistics, the precautionary principle, whatever. Okay, so what are statistical reforms? So the main reform that we had in the last decades was the so-called movement of evidence hierarchies inaugurated by Sackett in Canada in the 90s. And we saw that evidence studies have been developed in order to help doctors separate the wheat from the chaff. So evidence studies, I'm not talking about this anymore because it has been discussed thoroughly during the workshop. What I mean, the gist of evidence studies is to impose a credit system for evidence quality, and also to impose penalties for lower than good evidence, than average evidence, or imposing a credit system and then rankings. Now we have kind of biases. And we have these rankings according to which some piece of evidence should trump, for instance, RCTs should trump observational studies. However, as it was in Oxford in 2018, I remember Carl Hennigan, the director of the Center for Evidence-Based Medicine, saying, all things considered, medical evidence was better before the emergence of EBM, so the evidence-based medicine paradigm, than now. This is paradoxical. Is it paradoxical? Why? And maybe not. So the point is that standards of evidence may be exploited by interested agency. For instance, if low p-values are the precondition for publication, then people may engage in not finding the truth, but finding p-values, so selective reporting. So they conduct many low-power studies, and then they select the ones that have a significant, statistical significant results, so a low p-value. This leads to p-value, p-hacking. P-hacking plus noise produce the reproducibility crisis. For instance, we have here a high-power study on the left and a low-power study on the right. Generally, high-power studies are those with a large sample, and low-power studies are those with a small sample. If you can see that the two Gaussians representing the two hypotheses, the null hypothesis, the alternative hypothesis, in the low-power study almost overlap. So you can see that the ratio of true to false positives and true to false negatives is almost the same in the two, in the low power setting, whereas here the ratios differ substantially. What does this mean? It means that a low power study tends to act as a randomizer. The lower the power of a given study, the less discriminatory is the study with respect to the two alternative hypotheses. The two probability distributions representing the probability of the data, given the two alternative hypotheses, tend to overlap. The instrument, for instance, a particular trial, tend to behave like a randomizer, that is, to deliver true and false positives or negatives at almost the same rate. So when applied to the scientific process more generally, the result of all these hypothesis tests is a flow of noisy claims, which bear only a weak relation to reality, but which attain statistical significance. So p-hacking and other selective reporting practices are not only a problem for the reliability of single studies, but also because they bias the population of available studies one samples from for carrying out meta-analysis. So also here, scientific reforms, advanced solutions, pro-registration of studies, detection tools, funnel plots, p-distribution analysis, lowering the p-value. So the threshold should be, instead of 0.01, 0.001 or 0.0001. But this makes no sense because, and there's been a huge debate on that, because the larger the study, the higher the probability that you reach a low p-value. So one should consider the intrinsic noise of the population before saying that this is really a significant result. And then maybe changing the decision tool. Stop with hypothesis testing, let's go on with Bayesian analysis, or frequentists now go for confidence intervals rather than p-values, etc., etc. So here are an example of Bayesian statistics instead of frequentist statistics. This is a funnel plot for detecting bias in meta-analysis. You can see that there is here there is no asymmetry. Here there is an asymmetry of the location of studies. The higher they are, the more power they have. Let's say the larger the sample, and so they are presumed to get closer to the true value of theta of the population. And since the smaller studies are scattered around the true value, one thinks that they scatter around randomly both right and left at the same in the same proportion. But here we can see that there is a higher proportion, let's say, in the positive side. So there is a suspicion of bias of selective reporting. Here's a PQ value. I will explain it later on, so let's skip it. So what about pre-registration? I will focus on pre-registration because this is now the common requirement. So the mere effect of sampling error on the distribution of results of identical replications may provide the default explanation for the observed inconsistency of results across studies investigating the same hypothesis. So Sen and Goodman, for instance, they've said that there is no reproducibility crisis. It's just that by the very fact that there is random error, one study can detect an effect, and the other, the second replication may not detect it just because of the random error. However, it is precisely the awareness of biasing incentives that steers the debate about inconsistent replications towards the strategic forces driving knowledge production. So Ben Goldacre, the author of Bad Pharma, says, because we are detecting small differences between one treatment and another, so here the possibility of random error is, or random effect is high, we need to be perfectly hygienic with our data. So we spend all this money to exclude bias by randomizing, random allocation, blinding, etc. And then after spending millions, hundreds of millions, billions of dollars every year on doing randomized trials, specifically to exclude bias, we allow all that bias and error to flood right back in because we allow a half of all trials to be routinely and illegally withheld from doctors, researchers, and patients. It is insane. And so he developed this all trials campaign. It's also very nice. I mean, if you look at the website, there is a play the game, run clinical trials for yourself, choose which ones to publish, see how withholding results skews the evidence. Then the unreported trial of the week. So this is a blame game, etc. And around half of clinical trials have never been reported. This is the story of the campaign to find them and to fix medicine. So my question is, I think this is important. I don't think that this fixes the issue. So p-hacking, hacking, and the garden of forking paths. All trials campaign, as other analogous initiatives like Sense About Science, Open Science Foundation, etc., offer a solution, but shift the problem one step back or forward. So strategic behavior would move back from study selection to sample selection or forward to data analysis. And nota bene that the industry is already sabotaging it by involving patients group, etc. And also there are studies that show that the discrepancy from registered to published trials often goes unnoticed. So, I mean, okay, it may work, but it's a contributory process, but it's not the solution. So now let's dive into the real problem, which is behind the fact that no scientific reform will ever solve the problem forever. Smaldino and McAreef laconically note, statisticians have been publishing guidelines for decades and insist that issuing guidelines is of little help if we do not remove the bad incentives, which increase the chance of biased results. Indeed, one should not forget the famous Goodhart's law. When a measure becomes a target, it ceases to be a good measure. It's a little fish here. This phenomenon is also labeled as goal displacement by Teaser and colleagues and relates to the adaptive capacity to transform any evidence standard in a fake signal. Once a metric is established as an indicator of performance, efforts are put on showing that the metric is satisfied, rather than in actually achieving the indicator-related goal. So fake signals. The adoption of any quantitative social indicator to elect individuals to specific appointments will inevitably generate a competition towards positive signaling, where low types, that is, agents who do not intrinsically possess the required quality, may be incentivized to false signaling, that is, to show signals that indicate possession of such quality, thus deteriorating the same processes that the indicator was intended to regulate. A similar phenomenon has, for instance, occurred in relation to using the number of patents as indicators of research excellence for ranking research institutions and universities. As Mariana Mazzucato points out, this led to an inflation of patent requests for inventions of dubious novelty, for instance, generally slight variations on already patented inventions. Now, a case study on goal displacement. This is a study that we I'm speaking together with a couple of colleagues at the economics department, we carried out during the COVID case. So it's about conflicting evidence and conflicts of interest during the COVID pandemic regarding especially hydroxychloroquine. So this is a joint work with Nicola Matteucci, who's a political economist, and Matteo Picchio, who's an econometrician. So about hydroxychloroquine. So this is an all-purpose drug. First patent in 1951 in the US, whose original patent expired in 1968, and the drug is open to generic production in most jurisdictions. Hydroxychloroquine was covered by the US patent Rana. The inventor was Sare. In the less developed countries, the retail and subsidized prices of HCQ may reach minimum levels of few US dollars, or even dollar fractions per package. So the HCQ drug market features a non-monopolistic competitive setting with abundant quantities and affordable retail prices. Conversely, more recent or new drugs covered by valid patents have been developed or authorized as repurposed drugs since 2020 for COVID-19 treatment. In this context, the conflict of interests are expected to incentivize the replacement of out-of-patient drugs, such as hydroxychloroquine, with newer and more remunerative drugs or prophylactic alternatives, covered by effective intellectual property rights, molecular patents, software copyrights, etc. Why we chose this case? First, HCQ was a highly controversial therapeutic option against COVID-19. The controversy over HCQ for the treatment of COVID-19 led to the production of a considerable number of studies and an intense scientific debate with political implications. So it was not only about the truth of whether HCQ was effective, but there were a lot of economic and political factors involved. The evidence about efficacy and safety of HCQ for the therapeutic treatment of COVID-19 is characterized by strong inconsistencies across studies. And we wanted to test whether our COI construct could at least partially explain such variance of results, once other clinical and methodological variables have been taken into account. 3, a considerable number of the published studies failed to show any efficacy by HCQ, which is at odds with a notorious bias for statistically significant results affecting our publication systems, so-called publication bias. Now, we were a little bit astonished to see that there were so many highbrow journals like New England Journal of Medicine, The Lancet, publishing studies saying, look, HCQ doesn't work. Okay, I mean, it's really against the odds, okay? So what we used meta-regression and p-curve analysis to analyze this population of studies. Just a few words on p-hacking for the people that don't know p-hacking or the p-curve. This has been developed by Simon Schoen and Nelson and colleagues. P-hacking is considered as a self-serving choice of data analysis in order to increase the chance of obtaining statistically significant results. So there is an exploitation of data ambiguity, which outliers to exclude, which measures to use, etc. I mean, everything is, let's say, oriented to obtain significant results. And so evidence may be selected or whatever. So by definition, when an effect is non-existent, then A p-value lower than 0.05 will occur 5% of the times. A p-value of 0.04 will occur 4% of the times, and so on. Hence, a p-value between 0.5 and 0.4 will occur 1% of the time. A p-value between 0.4 and 0.3 will also occur 1% of the time, etc. So the distribution of p-values in a sample of unbiased studies under the hypothesis of no effect is uniform. Okay, so the upper curve is a situation where there is no effect, but let's say studies, I mean, the population has some studies that have, there is a p-hacking in this population, in the sense that some studies are published which show that right just before the 0.05 threshold value, there are more studies than ever. In this lower curve, instead, there is an effect, which is showed by the fact that the closer to the zero significance level, the greater the amount of population of studies in the population. The frequency here means the frequency of studies in the population. If there is an effect, then one would expect that there are more studies grouped around, I mean, towards the zero significance level, in the sense that they go away from the null hypothesis. And so they go towards the alternative hypothesis. And so there is a greater amount of studies which show a significant result. It means lower and lower. Okay. So what happened in our sample of studies? This is the p-distribution curve in our sample of 100 studies, more or less. So we have a lot of studies here, a lot of studies in close to the zero value and between 0 and 0.05, and then a uniform distribution. So it seems that there is no p-hacking, and then there is an effect that HCQ indeed works. Then we wanted to see whether there was, let's say, an explanation for the variance of these studies, because there were anyway a lot of studies also showing no significance. And so Nicola Matteucci developed this operationalization of conflict of interest. It was really complicated because he had to read all the conflicts of interest, looking at the EGM forms for conflicts of interest for all the authors of all the studies. And this was no conflicts of interest if there was no funding or competing interest, competing in a general sense, not only financial. And also if there was funding and competing interest, then we considered a conflict of interest of level 2. And so we can see here that when there is no conflict of interest, most of the studies are here, I mean, close to zero. When there is a medium conflict of interest, we see a curve, but it's less peaked. When there is conflict of interest 2, then there is a uniform distribution. It's bimodal, and it's even going upwards. I mean, it's not statistically significant, but it hits the eye. So there are two hypotheses for reading these results. These are exploratory results, of course. P-hacking curve are not hypothesis testing. So either HCQ is not effective, and the high COI studies faithfully reflect the situation, while the low COI studies are positively biased. In this case, the P-curve should show the typical bump just before 0.05 significance level in the subsample of the COI studies without conflict of interest. Sorry. Or HCQ is effective and the high COI studies are negatively biased, so there is a reverse p-hacking, while the COI studies faithfully reflect the situation. So the point is that either the people that have a conflict of interest faithfully reflect the situation, or the other ones. But in case the studies without any bias would, let's say, positively hack the results, then we should see a bump just before the 0.05. But this is not what we see. So if the COI is 0, the curve is really a no p-hacking curve. I mean, this is the sub-interval between 0 and the 0.05. There is no bump before 0.5. It's really going upwards towards a J curve. Okay. And then we did another study on also on HCQ, but on early treatment, because this is the time interval where doctors say that it works most, et cetera, et cetera. And we had these results. So that is that we have an asymmetry in RCT studies, and there is a negative, there is adverse selection against effectiveness of RCQ. And it happens that the COI overlaps with the research design. So RCT studies generally have a high COI, observational studies have a low COI. So one could think, okay, it depends on the research study. No. If you can see in figure 3, shows a mild asymmetry in the right tail, denoting the presence of few studies with high standard error, delivering a very strong effect size. As visible from panel C, these belong to the subsample of non-RCT studies. You can see here, these belong to the subsample of non-RCT studies. Focusing on graph B, we detect a strong concentration of the effect size around 0. So all the studies are here. I mean, this is not even a normal funnel plot for, let's say, a group of RCTs. This could be a signal of selective reporting issues, specifically an adverse selection for studies showing efficacy in the RCT subgroup. So these are the statistical results. Maybe I can just skip them. The results speak for selective reporting in the subgroup of studies affected by strong conflict of interest, which happens to coincide with a group of RCT studies. Now, although a common strategy adopted in evaluating conflicting evidence in medicine is to adopt a sort of lexicographic decision rule and prefer experimental over observational evidence, these results show that this may not be always advisable in contexts affected by conflicts of interest. Since RCTs get the precedence in such contexts, strategic agents will preferably decide to choose this sort of research design in order to get their message through. In this case, a genuine effect and no evidence of positive selection is present in the subset of observational studies, whereas RCT studies, preponderantly affected by COI, show no effect, but at the same time suggest issues of adverse selective reporting. So the former evidence, the observational evidence, should be more relied upon rather than the latter, the experimental evidence, because of these strategic forces at play. So let's see how a strategic view can be incorporated in, our approach to evidence in medicine. Philosophers and scientists have been advocating for a more comprehensive view of evidence, which may incorporate strategic issues explicitly and keep track of them in the inferential process. Okay, so I remember a very nice talk by Jacob Stegenga for the Peter Sowerby chair. He advances the skeptical view about the effectiveness of modern medicine because of the noise, bias issues that I explained also here. And Gell-Mann, was Andrew Gell-Mann, the great statistician, pleads for a direction towards Bayesian inference, in which data are combined with prior information. And prior information is not only the prior of the probability of the hypothesis, the substantive hypothesis, it's also prior about the higher order dimensions of the evidence. So in this case, for instance, the prior expectation that newly studied effects tend to be small, or that there can be bias, or that there is noise, or whatever. So some also, let's say, advanced hierarchical based approach where these hyperparameters, like from which population of means, in the sense of different populations of means, this mean come from, etc., so that we can have we can structure our uncertainty and our skepticism. I advocated for an expert system where all these pieces of evidence can be brought together, evidential pluralism, but where we have also this kind of reliability node which incorporate this kind of concerns about the reliability of evidence. So evidence gathering disclosure as a signaling game. So we enter here into the game theoretic approach to evidence. The theoretical research proposes that honest signaling behavior from a biased sender results from the receiver's sophistication in evaluating the provided evidence. So there is a skepticism effect. I am the drug industry, but if I know that the drug agency is very sophisticated and has the time to read all the documentation and to get through the trials, etc., then I have to be honest in giving in carrying out the trials, in giving the evidence, etc. Is this so? So increased research efforts and full disclosure of the acquired information becomes a means to signal honesty. So it is impossible to show everything. You know that the documentation for approving a drug can be of thousands of pages. We know that there will never be complete stuff reading all the pages. And so there is a problem of opacity. But of course, there are issues of reputation, of liability that can counteract this. So an upshot of this outcome, theoretical outcome, is that under the assumption that the responsible authority is sufficiently sophisticated in evaluating the evidence provided by the industry, observable testing, as advocated, for instance, in all the trial campaign, may be less than optimal policy. So the game theoretic guys say, look, you don't need to have total transparency. The point is that if the sender, the knowledge producer, knows that there is sophistication on the part of the receiver, he may indeed put more effort in doing more research in order to prove, and maybe delivers you only the best evidence that he collects. So he engages in selective reporting. Still, he has to do more research in order to deliver you this better evidence. Alternatively, or additionally, the receiver sophistication costs may also counteract bias, and they can be reputation costs, financial costs for liability, for class act actions, and so forth, and so on and so forth. Okay. So there are also this kind of, in this literature, there are also this kind of mathematical exercises. This is a very interesting paper, but very technical, that shows that, for instance, depending on the form, on the functional form of the noise, selective reporting can be beneficial for the receiver. And this, I mean, is done by using order statistics. It's nothing, I mean, nothing good. So, however, this was the theoretical research in the exchange of evidence between a knowledge producer and a decision maker. But the empirical research, let's say, didn't establish this theoretical result very firmly. For instance, the take-home message that honest signaling may be predicted as a function of the receiver's sophistication seems to be contradicted by empirical evidence. These studies show that subjects do not discount enough for the conflict of the incentives of the advisors. Advisor means the knowledge producer, the one that gives you the evidence. Also, investors do not fully appreciate the extent of manipulations, thus incentivizing companies to distort data. This happens also in finance. Analogously, Zingales and Dick show that investors do not discount the amount of distortion produced by firms in the counting data, leading to equilibria where firms engage in manipulative behavior rather than honest signaling. So there is this opaque area where even if I'm a sophisticated receiver and I know your interests and your conflicts of interest, still I don't discount enough for your manipulative efforts. And it seems that the manipulators know that and so exploit this. So another issue is that most of the theoretical analysis of evidence disclosure between knowledge producer and decision maker assume utility misalignment. So they say, okay, the big pharma is bad pharma, they want to manipulate, but the regulators are the regulators, they are the ones that want to safeguard our health. Is this true? or better, the decision-maker utilities are aligned with those, of the end consumers. But this is not at all guaranteed. I mean, there are self-interests also on the side of the agents and the officers of the World Health Organization or FDA or any agency. I mean, they are humans after all. And there is also the huge chapter of the regulatory capture. So if used for mechanism design, this kind of games that I've been talking about might fail to produce relevant benefits. Let's look at the moral collapse of Cochrane as a case in point. So the reputation for, you might know it, but just to recollect it, for unbiasedness and impartial watchdog of this institution has been recently severely compromised by a heated controversy regarding the safety of papillomavirus vaccines. Peter Goethe, who was one of the father founding of the Cochrane in Denmark, and two other colleagues published an article criticizing a Cochrane meta-analysis for severe flaws and biases. Instead of trying to justify themselves for allowing the publication of such flawed study, notwithstanding the warnings that Goethe himself had repeatedly pronounced, the top hierarchies of Cochrane, ostensibly confusing the cause with the effect, expelled Goethe. So instead of saying, okay, look at this meta-analysis, maybe it's wrong. No, let's expel Goethe from the organization on grounds of publicly displaying scientific dissent, not because he made a false meta-analysis of this meta-analysis, but because he had publicly displayed scientific dissent, thereby bringing the Cochrane into disrepute. How sad. John Ioannidis wrote a letter to the Danish Minister of Health condemning Goethe's dismissal from the Nordic Cochrane Center. Peter is undoubtedly a giant, one of the greatest scientists of our times. I believe that basic respect for scientific discourse requires that you do not eliminate your opponents through administrative mechanisms. So there was 3,500 scientists signing this letter, etc. Nothing helped. So we come here to the topic of scientific lobbyism. Here we have, for instance, a couple of books on the topic. I especially suggest to read Le Gardien de la Raison, which explains how the industry systematically lobby not only at the on politicians, but of course also on researchers, scientists, social media. So you have the social platforms. They pay or they retaliate people, etc., in a systematic way, pervasive, which we do not know about. This is Andrea Saltelli, who is an expert in scientific lobbyism, and he is a dear friend of mine, so I want to advertise his book coming soon, The Politics of Modeling, who exactly speaks about this interaction between scientific lobbyism and models, but in general, political or economic or whatever incentives embedded in the mathematical or statistical models that we use in science and in evidence evaluation. So, is the regulation a possibility? So Rice argues in favor of more private initiative and responsibility in the market for drugs. Patents for drugs should largely be abolished, and drug regulation by government bodies such as FDA or whatever should be abolished. The former claim, I mean, it's really provocative, but I'm thinking that it's not so far from... the former claim is based on previous work concerning the efficiency and legitimacy of the patenting system, the latter on a number of considerations which suggest that drug regulation is epistemically and morally deficient. I favor the regulation of the evidence market based on all the above. 1, strategic behavior sidesteps evidential standards and norms of admissibility. 2, even sophisticated receivers tend to not sufficiently discount for such distorting factors. 3, delegating decisions about drug approval to not directly concerned people who may be captured widens the information asymmetry and does not allow consumers to take informed decisions. So the only alternative solution is that loss of reputation, liability costs, or other forces guide the strategic behavior of knowledge producers with vested interests. Conclusion: evidence collection, interpretation, and disclosure is an epistemic game. Awareness of private information and personal incentives by scientists have resulted in developing evidence standards, admissibility criteria, and all the things I talked about. However, these statistical decision rules as mechanism design are always behind, let's say, the sidestepping work of the researchers or the industry, because they create themselves game-specific incentives which may backfire. So there is an evolutionary game. So let the invisible hand guide the evidence market. Here are two papers where I talk about these things, and the second is forthcoming. So if you like me to send it to you, I'm happy to share it. I'm also sharing this news. We founded the Center for Philosophy, Science and Policy in Ancona, where I'm affiliated together with Alexander Goodhart, and anyone who's interested in joining us, participating, is welcome. There are many of you here who are already members of the center, and I hope to see you in Ancona. Thank you. Now I stop to share. Okay.
Thank you so much, Barbara. Can you hear me? Yeah, I think you can.
Yes.
Questions? We'll just get straight into it. Okay, there's a few. Start up here and then I'll come to you.
Thank you. I think it was a fascinating talk and very interesting methodology for looking at this. I wondered, just the conflict of interest data and that correlation with the negative signal in hydroxychloroquine. Do you consider that the conflict of interest may just correlate with status as investigators, which may again show why it's correlation with large-scale randomized controlled trials, and that therefore there's a significant confounding factor in that? And now also, just on a related point there, because obviously we're talking in that case, the New England Journal paper that showed no effect for hydroxychloroquine was by the recovery trial in the UK, which also showed a positive effect for dexamethasone, which was another low-cost medication, which would somewhat go against the thesis that they were captured by the market. Anyway, but fascinating.
Thank you. Regarding the first question, the status in what sense? Sorry, the status of researcher as an academic affiliation level, or?
Yeah, I suppose that the very high-profile researchers are likely to have funding from lots of different bodies and likely to have interactions with pharmaceutical companies as well. And therefore, that may be a confounding factor. That might be higher quality data coming out in that instance. I mean, certainly.
Well, how can this be? I mean, we had a lot of variables there. We checked for conflicts of interest and also affiliations and also the level of the journal, et cetera, et cetera. And the main explanatory factor was conflict of interest. But in what sense, what would be the causal structure according to which the affiliation is a confounder? I mean, in order to be a confounder, it should be either, a common cause of doing an RCT and manipulating or not manipulating or being selected for that. I don't see how this can be a confounder.
Okay, so RCT is very expensive, very hard to do, very difficult to get funding for. You are more likely to get funding if you have high status within any given discipline or specialty. And I would suggest you're quite more likely, I don't know, but I would suggest that you're more likely to have what might be termed conflicts of interest if you are high status and have had lots of funding.
But this is just an overlap, and this wouldn't, let's say, deflate the relationship between conflict of interest and adverse selection against HCQ, because the group of studies with COI overlaps with RCTs and displays adverse selection against HCQ. So it's not a confounder, it's just an overlap between RCTs and you can see it also from the fact that there is a concentration around the zero value, which is abnormal, and there is no symmetry in the RCT group. So the RCT group, which is also overlaps with the COI, displays an asymmetry. in the study sample. But let's go to the second question, which was, I don't remember now.
Was simply that the suggestion therefore is that they've been in some way captured and that's influencing the publication or the production of these negative signals. But the main study that showed the negative finding around hydroxychloroquine was the recovery trial, which also showed a positive finding for dexamethasone, which was a similar low cost, pennies intervention.
I don't remember. I mean, I remember the recovery trial. I remember that the recovery trial was strongly biased methodologically because they gave them 1,200 milligram dosage, which is fourfolds the therapeutic window for regarding hydroxychloroquine, which didn't know. The recovery trial didn't show that the HCQ was not effective. It even showed that it was unsafe. I worked with doctors. The reason why we chose this study is also that I was called by doctors in Italy to defend them, and we had a trial against the Italian drug agency because they wanted to abolish the use of hydroxychloroquine for COVID in a time where there was nothing else. So it was also from a risk-benefit analysis. I mean, it's better this than death. But apart from that, the recovery trial gave patients fourfold the therapeutic window of dosage. So it was intentionally giving them a toxic dosage of hydroxychloroquine. And this was valid also for ivermectin. And I can tell, I mean, we won the trial. So at least in Italy, they didn't we could further prescribe hydroxychloroquine, and there were doctors that cured oncologic elderly patients like 90 years old with cancer that were cured from COVID.
So I can imagine this back and forth could keep going, but I'm going to interrupt because I think there's further questions. So there's one here and then over.
Well, thanks a lot for your very interesting talk. I really liked this approach to analyzing reported p-values. And of course, I guess we agree that p-values are random variables, so that the distribution that you observe may also result from just random sampling and not from there being a conflict of interest. And I would like to ask you, because in 2020, there was laboratory research conducted in Germany by Hoffman et al. 2020 that showed that there are two separate mechanisms by which SARS-CoV-2 can enter cells. And that study explained why we observed that hydroxychloroquine is effective in kidney cells. Because the mechanism by which SARS-CoV-2 entered kidney cells relied on an enzyme called catepsin L. And that enzyme is dependent on acidity of the environment. And hydroxychloroquine changed the acidity of the environment so that hydroxychloroquine disabled that enzyme. While in the lung cells, SARS-CoV-2 entered cells by different mechanisms. And for this reason, hydroxychloroquine could not be effective to treat the spread of the virus or to stop the spread of the virus in kidney cell, in lung cells. And since we know that COVID is primarily a lung disease, on this ground, it was argued that hydroxychloroquine could not be effective. And in the paper co-authored with Adrian Stenzel, so it's Majash and Stenzel 2022, we argued that this study delivered negative mechanistic evidence, so that we knew that hydroxychloroquine cannot be effective in humans
It's true and false. May I? It's true that it doesn't work at the level of the lungs. That's why the doctor said that we should prescribe it as soon as possible. The mechanism of the hydroxychloroquine worked at the level of not letting the virus entering into the cell when the infection was oral. So, and so that's the mechanism, and there is a paper on that, so I also can explain a little bit more at length. The reason why we won the trial is also that, apart from this mechanistic evidence, which tells that it worked in early stages, and indeed the recovery trial tended to give it at a stage where it was not possible for it to work, as you say. COVID is not essentially a lung disease. COVID is an inflammatory disease that produces cytokine storm. And hydroxychloroquine has been used for years against not only malaria as an antiparasite, but also against rheumatoid arthritis, which is a chronic inflammatory disease. So also from just a mechanistic plausibility, let's say, consideration, one could have considered that in a risk-benefit analysis, one should have given hydroxychloroquine everywhere, like they did in India, for instance, where they didn't have the death toll that we had in Italy. Another issue is that if you give hydroxychloroquine or whatever medication, ivermectin, whatever, for chronic diseases, that is for years, how can you say that it has no positive risk-benefit profile? I mean, you would use hydroxychloroquine for COVID for at most 1 week, 10 days. This is what the doctor said to me, and it worked. And I mean, this was a medicine that was used for years, since the 50s for arthritis rheumatoid, for years for the patient in a chronic condition. That's so. I mean, considering all the things and considering all the dimensions of the problem and of the evidence, I see a problem there. Of course, in Italy, we might be more concerned because we had one of the highest death tolls for COVID.
Thanks. Okay, so we've got 5 minutes, so let's make them quick.
You said at the end of your talk that you would like to deregulate the evidence market. And could you say a bit more about what that means, and especially the demand side? Who would be the buyers of the evidence once regulators drop out of the picture? And doesn't that lead to a huge amount of bad studies that just exacerbate the noise, and few people will be able to read all that?
I think, of course, it's tricky. I mean, we have to think about how, and I'm seeing a lot of opposition to that. I think it's a utopia, but it cannot be worse than that now. It cannot be worse than that. In my ERC project, I worked with drug agencies all across Europe, the Italian drug agency, the German drug agency, the France drug agency, etc. As COVID emerged, they were all shut down. I couldn't contact anyone. It was all shut down. Before that, I had already a hint of this regulatory capture, which is not necessarily financial corruption. I mean, these people, they are these black men from the big pharma, they become friends. I mean, when I worked with these agencies and these officers, they were talking about their colleagues from the pharma industry as real friends. They have been knowing for years. They go to dinner together, etc. They are friends. And so it's no longer monitoring, a well-functioning monitoring system. I mean, we have to find a different way. And if we look at for instance, at the market of food supplements or vitamins, etc., the people just inform themselves. They become proactive, they empower themselves, they look at the information, they look at the excipients, etc., and say, okay, this might work for me, or might not. And then if they see that the product is not very good, they don't buy it. They might be also, let's say, self-regulating bodies, like we tell as industries in the fat supplement which are the processes according to which the element is more pure or whatever. I mean, it has to be thought of, but we have to think about it. Might be a possible way to go.
Great. Okay, so we had 2 minutes. Quick question. Yeah.
Okay, thanks. So health technology assessment is a subfield of implementation science, and it is the one field that's trying to deal with this, all of these that you're presenting. So do you know about Great Qual approach, and what do you think of it, especially because it has developed a variety of components, and especially dissemination bias? And do you know about Elicit that's being developed by the Epi Center at UCL? Because, you know, he brought out large language models into evidence synthesizing, like systematic reviews. So what are your thoughts about those two tools?
Oh,
thank you.
I know the first one. I don't know the second one, so I am happy to hear about that. I will have to look at it. The grade is fantastic. I mean, it's a little bit, don't take me wrong, parasitic, in the sense that it's always relying on the evidence hierarchies and the lexicographic approach. So let's first have the internal validity, and then external validity, and then look at the bias, and then put this together, etc., etc. I mean, they are in some sense implementing what I'm saying. Indeed, mine is a philosophical talk. It's not about a methodology. Anyway, the point is, for instance, when I looked at their papers, I had issues regarding evidence for risk, because of course
it's 18 hours.
Risk comes with anecdotal evidence, and then I mean, you discover risk, you detect risk more than testing it. So in the I don't remember now the exact, I think that's I mean, one of their papers dealing with changing the hierarchy of the ranking of the evidence. The evidence about risk would systematically be put lower on the ledger, and so if you make a benefit risk assessment, the benefit would always have a higher weighting. I mean, systematically, because generally evidence from benefit comes from clinical trials, and so it will weight necessarily systematically more than the evidence for risks. But this is the only. I mean, it can be corrected, so it's just. I don't know the other. Thank you for telling me. I will have a look. Thank you.
Great. So I think that's all we have time for. Thank you so much, Barbara. And I'm sorry you can't be here, but that was great.
Thank you. Thanks to you all. It was really nice. Nice to see you.